evolutionary genomics is one area where sort of learning complex models arises naturally. The basic sort of phylogeny reconstruction problem is you sequence some gene from a bunch of different species, and you reconstruct a tree that sort of represent how they're kind of related by evolution. But as soon as you sort of start looking at whole genomes or big portions of genomes, you get sort of a more complicated picture, where different genes might be telling you different stories. I'm going to tell you a little bit about two sort of biological phenomena that use this one, gene duplication, and one is incomplete lineage sorting. But in both cases, you have to sort of come up with a more complicated model to try to sort of capture these phenomena, and then you have to develop some inference methods that are tailored for them. In particular, you get sort of these very large data sets where you have to worry also about sort of computational efficiency of the methods and so forth. So let me just start very quickly by giving you some background on what estimating G-tree is, and then I'll get into these two more advanced applications. Okay, so this is the basic problem. I don't assume that you know anything about this. The problem is you have a bunch of species. These are some Darwin's finches, and you'd like to know how they're related to each other. And so what you do is you sequence some DNA, and you get a table like this. And so here each row is a different species, and each column is a different position in your DNA. Now, in this arrow, there's a lot of pre-processing that goes into this. You have to kind of align these sequences and make them look like this. And so as we can describe in this talk, we have insertions and deletions. These sort of happen during evolution also, and you sort of have to correct for them by aligning these sequences between removing these insertions. But once you have a table like this, the basic idea is that the more sort of similar, the closer the species are in the tree of life, the more similar their sequences are, because there's been sort of fewer substitutions along the way between them. And then from this, you're trying to reconstruct this evolutionary tree here, where the root of the tree is this sort of common ancestor of all these species. Each branching corresponds to a speciation event, with some new species being created. Think of a bunch of birds moving to a different island, is trying to reproduce independently. And then at the tips of these this tree, you have your current species. And there's also some extinctions, which I'm not showing. The lineage is an end. This is the goal. And so to turn this into a reasonable statistical problem, you have to sort of model what this process is of evolution of sequences on the tree. And that's a very well understood sort of problem. Here's a sort of standard way to do this in evolutionary biology. If I look at just one column for now, so these are different species. This is one position in my DNA. I'm going to imagine this is my evolutionary tree, which I don't know, but it exists. Each branching, again, is a speciation event. And so at the root here, I've got my common ancestor, and I'm going to imagine that I have a latent graphical model on this tree. That is, at the root of the tree, there is a distribution for the state of. I'm not showing you the ancestor. These are all contemporary species, but there's a probability distribution for the state of ACGT in that position for the ancestor. And then along each of these branches, you have a conditional probability distribution that tells you what the probability that given the ancestor is A, that this descendant inherited A or J or C. So here in this case, I might go from A to A, or from A to G, depending on these probability distributions. And then I repeat this independently on these edges. And eventually I get a letter for each of my contemporary species, which are the leaves of my tree. And that gives me my columns. These are the first 3 rows of these columns. But then I'm going to imagine that each position in my DNA is just a bunch of independent samples from the same model, the same tree, from the same parameters. And so I just get k IID columns that are generated by this process. And my goal is to recover the tree and potentially reduce the parameters on the edges that is unknown. So there's lots of ways to do this. One particularly simple way to do this, which is useful here because it's sort of computationally efficient, is to think of your tree as a metric. Right? I said one of the basic intuition here is the further apart you are in the tree, the more mutations is going to be in between. So there's a sort of metric hiding behind. There's a lot of notation here. I'm not going to use any of this. You don't even know what it is. But just look at the picture down here. I'm going to think of my tree. So first, forget about the root. I've got some unrooted tree here. Again, the leaves are my current species. And I'm going to imagine that on each edge, I have some positive number, some positive weight. Think of it as the expected number of mutations per site along this branch of the tree. And now I can define a metric on the leaves of the tree by just, it's a tree, so between any two leaves there's a single path, and I can sum the weights, and then that gives me, I do this for every pair of leaves, and that gives me this matrix. I'm going to call it the distance matrix. And that thing is a tree metric. It has the property that not only is it a metric, but if I give you a tree metric, there's some simple ways to recover the underlying tree from that distance matrix. And let me just show you very quickly how this works. The idea is to look at 4 species at a time. So if I have 4 species in my tree, and I don't care about the root, these are the 3 possible configurations. I care about, these are the same topology, but I care about the position of the leaves on these trees. The fact that dog is next to cat or dog is next to frog and so forth actually matters here. And so I've got 3 different ways of labeling this particular tree. And now if I give you a tree metric that comes from one of these, and you want to figure out which one you're looking at, you can use this thing called the 4-point condition. That is, I'm going to take the distance between A and B, I'm going to add the distance between C and D, I'm going to subtract AC, and I'm going to subtract BD. This is what I'm doing. Add, subtract. And then because of the additivity, right, the distance between 2 things is the sum of the weights, there's all these cancellations that happen here. And if I do this, and this happens to be the true topology, what I'm going to get here is minus twice the length of that middle edge. If I repeat the same thing on this topology, I'll get something negative. If I do the same thing on this topology, this time I'm going to get the same formula, so AB plus CD minus AC minus BD. This time I'm going to get, because of the cancellation, twice the number of the middle edge. If I do this on this one, I get 0. So in principle, I have a way to distinguish between which of these topologies my metric came from. And then I'm going to repeat this for every set of 4 species. And it turns out it's a little exercise. Think about it. Once you have all of them, it's like a little puzzle. You can put them together into one big tree that fits all of these smaller pieces. So there's some simple simplification. So that's one way of reconstructing a tree. Now, the one thing I haven't told you yet is what is the distance that I'm actually using. And for this, I have to go back to my genetic model. And so the natural thing to do here is to say, well, I've got all these pairs of all these sequences. I can just look at any pair and count how many differences there are between them. So that would give me some notion of distance. It turns out that's not an additive metric or a tree metric in general. The mutation rate changes from branch to branch, and so it creates distortion. That doesn't work. Instead, you have to come up with some other way of sort of modifying this idea to get you something that's additive on the tree. And one standard way to do this is called a log-dead distance. Okay, so if fxy here is the joint distribution of 2 leaves, x and y are 2 leaves in my tree, and if I look at the joint distribution of their positions, so how often do I see AA or GG or TC and so forth, that's going to be the 4x4 matrix. I take the determinant of that minus the log, and I call that my metric. And that turns out to work because of the Markovian structure here, this joint distribution of x and y, you can think of it really as a product of transition matrices. And so the determinant of this product becomes a product of determinants, and you take the log and you get a sum. This thing just sort of adds up over the edges, and you get a proper distance there. Okay, so this is based on the true model, and in practice what I do is I estimate it. I take the empirical frequencies of all the pairs that I observe, and that gives me an estimated true metric that I use to plug into my, for example, my quartet-based method trees as well. And so that gives you something a little bit more. It also tells you that this model that I've defined is actually so you get this log-dead distance, so that's a tree metric. And then if you go back to your model, you get the identifiability of the model. It's not obvious. If I have a tree and I tell you the distribution at the leaves of this tree is it the case that reconstructing the tree so that a unique solution, that turns out to be the case because of this tree metric idea. If I take two different trees, they're going to produce two different metrics because I can reconstruct the tree with this tree metric. And since I built this metric based on the distribution, I get that the tree itself, if I ignore the root, is identifiable to the distribution of the data I built. So another way that I can do, just quickly, to reconstruct the tree is to do maximum likelihood instead. So I've shown you sort of fast method, which is this tree metric-based idea. But you could also, once you have a model and it's identifiable, you could just maximize the likelihood of your data. I assume that each of my columns is independent. Computing the probability of observing a particular column on a fixed tree is something that you can do quickly. And then you find the best tree that maximizes the likelihood. That problem is NP hard, but people have developed a lot of heuristics in practice as well. And that's probably the most used method actually. So one thing you can show is that as the number of columns goes to infinity, this thing is guaranteed to converge to a true tree. So it's a consistent estimate. You can also get sample complexity balance. How many samples you need to get the correct tree, assuming that the branch lines are a certain bound, depth of a certain bound. And you can do the same thing for the distance quartet-based. So just a quick overview of this gene tree estimation problem. Okay, what I really want to talk about is what happens if I look at the full genome. So instead of just looking at sort of one piece of DNA each of my species, I got their entire genome with big portions of it. That's very common nowadays. You might think, well, okay, so that's sort of an easy problem. I'm just going to, so, you know, there's a first step here, which is not obvious. You cannot just take two genomes and put them side by side and just, you know, compare them 2 by 2. There's a lot of things that happen during evolution. Things move around and so forth. And so you have to do some of this pre-processing to get things to align. And so you can break up your genome into a bunch of smaller pieces. I'm going to call them gene, but in general it could be something else. So things that you can align across different species that they've inherited from a common ancestor. And I have to do this multiple sequence alignment thing so that, you know, I remove sort of the insertions and deletions. But eventually I have a bunch of genes that I've been aligning across all of my species. And from that information I'd like to reconstruct the species history, that evolutionary tree. And so you might think, well, let's just take all these genes and just, you know, put them into one big super gene of length m times k, say, if each one is length k. And then I'm just going to apply, say, maximum likelihood to reconstruct the tree out of this. And unfortunately, this doesn't work. Well, people do this in practice, and it works in some situations. But it doesn't work because it turns out that different genes can have different histories. And so to explain that, I have to kind of introduce these two different trees. The one that I ultimately care about, at least for the purposes of this talk, is this what's called the species tree. These are going to be these fat trees here. And they correspond to the actual evolutionary history of my species. And so the leaves here are my current species. The root is the ancestor, and each branching corresponds to a speciation event. Okay, so a new species being created. And I'm going to assume that this evolutionary history is actually a tree. Right? It might not be in some cases, but let's assume that the evolutionary history of my species is a tree. And now what is the other tree on the other side? So the thin trees are going to be the history of a particular gene. Okay, so if I take one gene and I look at one copy in one species, one copy in another species, and so forth, and I sort of trace back in time where they came from, ancestor to ancestor, eventually these lineages meet, and I get some tree there that I'm going to call the gene tree, and that's going to be the history of that particular gene. And if I look at different genes, I might get different histories. Okay, and so for various reasons that are sort of depicted here, you might end up with different genes having different trees, and that creates problems. Okay, so let me go specifically into one of them, which is called gene duplication and loss. Any questions so far? Okay, so what is gene duplication? Once in a while, because of mistakes in the sort of reproduction of DNA, you might get a whole section of DNA that gets duplicated. And so now your descendants have 2 copies. For example, I have 2 copies of a particular gene.
You can have more complicated things. Let's just assume that one gene sort of gets duplicated, and now I have 2 copies of it. I could also sometimes lose it, as I can go in the other direction. I could lose a piece of it. And now, again, my goal ultimately is I've got a bunch of gene trees that I've sort of reconstructed. My goal is to get the species tree out of it. Why is this sort of duplication and loss creating potentially different histories? So this is one case where something happened that created a different history. So again, the thin tree is the history of a particular gene. And now imagine that at some point here, okay, so there was first a speciation event. So I got sort of 2 lineages of my particular gene here, one for each of these species. But then there was a duplication where suddenly I have one of these species has 2 copies of it. This is what this is represented here. The dotted thing is like a separate lineage that corresponds to the second copy of my gene. Now there's another speciation event here, so each copy got reproduced, and the ancestors, the descendants got them. But then later on there was a loss here, so this particular lineage was loss. There was a speciation, and then there was a loss, and there was a loss. Okay, so a certain history of duplication and loss. And now what is the outcome of this? Right? I don't know any of this. Right? All I get to observe is what sort of happens at the tip. And if I reconstruct the history of this particular gene using my G-tree estimation method, I might get something that's wrong here, because in the species history, C is sort of sibling of B, whereas if you look at the history of this particular gene, your C is sort of sibling of D. Because of the particular history that happened with the duplication and losses, C sort of met the lineages from D first, and eventually the lineage from B that actually came from a different copy, but I didn't know that. Okay? So because of this thing, my particular gene might have a different history from the thing that I'm trying to reconstruct, which is the species history of my species. Yes, go ahead.
Sorry, when you explained this picture,
Yeah.
so does the explanation go through a simultaneous speciation event that both the species with 1 gene and 2 genes, they both speciate? Isn't that extremely rare?
There's no simultaneous speciation. These speciation happens sort of independently from one another, right? So the speciation that happened here is sort of independent from the speciation that might have happened down there. What I'm representing here is really just a duplication where some portion of DNA was sort of doubled, and that was sort of inherited by the whole population eventually. And now I've got sort of 2 copies of my gene in that one population.
I think your question is that's the speciation, the second from the top one. Right. Those guys all have these 2 copies.
Oh.
It's the same. It's one species that speciates.
Oh, this particular one here.
Yeah.
Yes. Right. So this is just representing 2 copies of the same gene that were both inherited by the descendants. And so there's a descendant here, there's a descendant there. Both of them get both copies. And I'm representing the actual lineage of where they came from in a tree, which has a different meaning than the tree over here. These are not speciations. Again, some of them are speciation, some of them are duplication. But the simultaneous part is just that one individual inherited the 2 copies of its ancestor. Yes?
Like this thing of some species suddenly duplicating a gene. I mean, you aren't thinking of this as a speciation event.
No. So this is just some, all right, so there's actually a whole population in there, and you're saying maybe some of them have the duplication, some of them don't. And so we imagine here that what ends up happening is because this duplication was somehow favorable in some way, because some new functions was developed or something, suddenly that sort of spread out through the entire population. I'm sort of hiding that level of detail there. Okay, so what we'd like to do is we get a collection of these genes. For each of our genes, we've reconstructed these trees, and now we'd like to reconstruct the entire species tree. And so for that, I have to sort of model what is the relationship between the species history and the particular gene lineage history that I have there. And what we'll do is something very simple. Right? So we'll imagine that it's just a branching process, that there is an unknown species tree that is just fixed. That one is not random. It's just the actual species history that exists. And then if I looking at one gene copy in one of these individuals, then as I follow these, you might have at a certain rate branching, where I have a duplication, and at a certain rate you might have losses. And these These might vary across different branches, but basically what happens in each branch, you have this branching process that duplicates and loses. And then once I reach one of these speciation events, each of these things gets split into two because all the copies get inherited by the descendants. And then I repeat the same process in each of my branches. I've got some duplications and losses that are happening at a certain rate. And now for a fixed species tree, eventually I get a random gene tree that's generated by this duplication loss process. And note that I could have several copies of some of these genes. And so what I really have is sort of more like a gene family. I've got a bunch of genes that sort of look the same. And unless I actually try to reconstruct their whole history, I don't actually know which one corresponds to which in different species. So I just have this gene family. And I'm going to do this for several gene families. And each one of them is going to be an IID sample from this process. Okay, so what I really have here is this sort of hierarchical model where there's this unknown species tree. For each of these, for each gene family, I get a random gene tree that's generated by this branching process. And then on top of that, for each of these gene tree, I don't actually get access to them directly. What I get is the genetic data. And so there's on top of that my first model, which was this DNA sequence model evolving on the gene trees that produces the actual genomic data that I've actually sequenced and sort of aligned and everything. And my goal is to reconstruct the species tree from this. Now, what they often do in practice is they sort of ignore the fact that it's a hierarchical model. They first, well, they don't ignore it, but they first reconstruct the trees from the genes themselves. And then from this collection, they get the species tree in some fashion. They, most of these methods sort of assume that the gene trees are actually correct. That is, in that first step, I actually reconstructed the true gene trees from the genetic data, and then from this collection I get the species tree. I'm going to make that assumption here for the sort of theoretical results, but it's an assumption. It's actually not true in practice. So this is sort of my simplified model where I ignore the sequence part. I just have a species tree that generates gene trees. From that collection of IID samples, I want to get back the species tree. And so how am I going to do this? And again, you want to do this in a way that's sort of computationally efficient. So one simple way is to go back to this idea of breaking things up into smaller pieces and putting them back together. That turns out to be a powerful way to solve a lot of these problems. Imagine again that what I'm going to do here is I'm going to look at 4 species at a time, so A, B, C, D, and for each of them, I'm going to look at, this here says, let's look at all the possible ways of choosing one copy from each. But just to simplify, let's imagine I just pick one random gene. So if there might be several genes in my species A, several gene copies in my species B, and so forth. Just pick one at random from A, one at random from C, one at random from D, assuming that there's at least one in each of them, which might not be the case. And then I'm going to repeat this. I'm going to construct the particular quartet history that corresponds to it. So if I choose one copy from each of these species, and I look at the restriction of my gene tree to those 4 guys, I get one of these 3 quartets. Okay, so that's my reconstruction for this particular gene family. And then I repeat the same thing for each of my gene families. For each one of them, I get one of these 3 possibilities. And then I just do a majority test. Which of these 3 happens most often? And then I call that version the most likely, the most common one in my data set. I sort of, that's my reconstructed quartet for that species. And then if I do this for every set of 4 species, and if I'm lucky, they all sort of fit together and I get a tree out of it. In practice, that's often not the case, but there are these sort of methods, one of them is called ASTRAL, that will take these quartets like this and try to find the best possible fit of a tree that sort of fits as many of those as possible in some sense. And that's the algorithm that we'll consider. As I said, it's sort of a variant of something called ASTRAL. That turns out to work, so it's sort of computationally efficient, and it proves the following. So one thing that I haven't said before, but that was open until recently, so there's been a lot of work on these various sort of phylogenomic kind of models in evolutionary biology. I'm going to talk about one that's very well studied in a second if I have time. But this duplication loss one is sort of less studied, and so particularly it was not known whether this model is even identifiable. That is if I take two different trees, do I necessarily get two different distribution of gene trees? And so this particular quartet-based majority method shows that indeed this is identifiable. The unrooted species tree topology is identifiable. And it also gives you a statistically consistent method. That is, if I get a large number of independent copies of my process, then in the limit, this particular method is guaranteed to reconstruct the true species, this true species history. Just to give you an idea of the proof, so why is it that from this I can get back the true history? And so we said before that it's enough, in order to reconstruct a tree like this, it's enough to look at just 4 species at a time. So if I get a correct reconstruction for every 4 species, then I can get the true tree out of it. So I suppose that I look at these 4 species here. Why does it work there? There's some computation to do, but there's sort of different cases that might happen. If I take one random copy from each, and I'm just going to follow backwards in time where they came from, one situation that happens is that if one of these coalesces backward in time, so if they merge backwards in time into one of these populations here, then automatically, if you think about it, you get the right quartet topology. What happens otherwise, this is where you have to start keeping track of everything. If that doesn't happen, then it means that all of these lineages sort of backwards in time meet in that ancestral population over here. And now there's different cases. If they all meet, if they all come from a different gene copy, then things are sort of symmetric. And each of the 3 topologies is sort of equally likely under this model. If not, then some things are good, some things are bad. If one from here and one from there chooses the same ancestor, then you get the wrong topology. So you have to start doing some accounting there of these different cases, and depending on how many different ancestral copies there are. And you have to be careful that you're also conditioning on the fact that there's going to be one in each and so forth. So you put all this together, and in the end, this all works out. That is, the most likely topology of one random copy from each is going to be the one that actually corresponds to the species history. And if you do this for every one, you get the entire species tree out of it. And so this will appear in Recon. We actually implemented this method and to see how it does compared to other methods. What you're seeing here is, so these are simulated data set on a real tree. So they took some 16 species tree. And then for different numbers of genes, so these are what the columns are, different rates of duplications and loss, which is what these rows are. What you get to see is how different methods, these are different colors or different methods here, how accurate they are. And so this particular scale here just counts how many edges are correctly reconstructed. So the lower it is, the better it is. And so our method here is ASTRAL, which is this yellow one, and it does fairly well compared to everything. These other methods are much more sort of combinatorial and much more kind of intensive. They take all of these trees and they kind of try to map them in a sort of combinatorial fashion to a species tree, and then they find one that fits well. And so this method, ASTRAL in particular, is going to scale much better if you look at larger scales of trees. Okay, so let me change gear and tell you about a different reason why, in the last 20 minutes, why you might get different genes telling you different stories. This one is called incomplete lineage sorting. It's modeled by something called the multi-species coalescence. So forget what you know about duplications and losses, but let's go back again to, I've got some species history that I want to reconstruct, and now I get different gene histories. And now what I'm going to take into account is the fact that there's sort of population genetic effects that can create discordances between different gene histories. And so in this case, what you want to do is sort of go backwards in time. And what you're seeing here is sort of different generations of your population inside one species. So these are the ancestors of these guys. And each dot here is a different individual. And now if I take one gene copy from here, one gene copy from there, and so forth, and I just going to keep track backwards in time where it came from, ancestor to ancestor, I get a lineage like this. And eventually these lineages are going to meet in some ancestral population. And then when they meet, I get a merging like this. That's called a coalescence, and so forth. And so I get a tree for this thing. And so this is the same idea as before. But now, what is incomplete lineage sorting is what's depicted here. When you have a lineage coming from two descendants here that are not actually meeting in that first ancestral population where they could have met. And it said it meets later on. When that happens, then this particular lineage in this case ended up meeting first with something that's less closely related in terms of species. So here C is more closely related to D in the gene history than it is to B, whereas in the species history, C is more closely related to B. And this is just because of this sort of random sorting of lineages that happens in populations. And so we're going to model this using the multi-species coalescence, which is, the coalescence is just this process where you have a bunch of lineages inside one population and each pair merges at rate 1. And so this is sort of what happens here. I've got this like discrete version of that here. And so if you have 3, then each pair merges at rate 1. Eventually, when there's 2, the remaining pair will rate at merge 1 and so forth. So you have, again, a stochastic process, and then you repeat this on each population, and you kind of stitch them together, the corresponding lineages in this one. And so that creates a distribution over gene trees. And again, you imagine that you get a bunch of IID copies from that, and your goal is to reconstruct the corresponding history. And so once you have a model like this, you can actually compute probabilities of different things happening. So this is human, chimp, gorilla. If you look at human, chimp, gorilla, right, so presumably chimp is more closely related to human than to gorilla. But if you look at different genes in those genomes, you'll see that there's a certain fraction of them, a non-trivial fraction of them, where it looks like chimp is more closely related to gorilla. And one possible reason is what happens here. And so that depends a lot on sort of the parameters of this ancestral population here. The shorter it is, or the larger the population is, the more likely you'll have this incomplete lineage sorting that will result eventually, potentially, in a discordant history, as is depicted here. And so if you normalize things in the right way, you get the sort of the length of this branch sort of affects the probability of observing these different things. And so now we have a model to compute things, and we can go back and do the same thing. That is, let me again ignore the fact that first I'm going to reconstruct trees from my sequences and then a species tree. Let's just assume that we get a collection of IID samples from the multi-species coalescent that are all correct. And then my goal is to reconstruct my species tree. These are called summary methods in this particular literature. And there's lots of different ways of doing this particular step. And so this is a much more developed area. There's been a lot of work in there in practical methods and also in theory to understand this particular process, which seems to play a role in many problems. And I'm going to look at one in particular that's used a lot in practice. So this is called NJST or ASTRAL. And about which very little is sort of understood in terms of their theoretical properties. Okay, so how does NJST work? It uses, again, this idea of thinking of my species history as a metric over the leaves, but then it's going to define a particular metric that doesn't look particularly intuitive at first. And so again, I've reconstructed a bunch of gene trees, and for each of those, I can look at a pair of species, say X and Y, and on my gene tree, so the thin one, I can count how many edges there are between these two species. So this is called the internode distance. And now if I get a different tree, I get a different distance. So here there's 3 edges between them. Here there's 2 edges between them. And now imagine that you just take all of your gene trees and average these numbers across all of your species. And I do this for every pair, and I get this internode distance matrix. Okay? And then from that, I'm just going to do the same trick that I did before. I reconstruct 8 quartet topology, then I put them all together. And it's not obvious why this should work. It looks like a sort of a hack, because, I mean, why is the number of edges have anything to do with the sort of the actual distance between things in the tree? But that turns out to work. And the intuition there is, again, if I look at one of these particular gene trees, if none of these sort of meet before this ancestral population, then because of the symmetries of the process, every possible topology is sort of the same, as the same probability. And in particular, what I'm going to end up getting is the, so remember that in your, what I really care about right, when I'm trying to reconstruct from my distance is this 4-point condition. And for every 4 pair, I look at this 4-point formula, and it tells me the length of that middle path. When that happens, each of them is sort of equally likely, and everything sort of just cancels out in the expected 4-point condition. Whereas if something like this happens, then you can convince yourself that you're always going to get something positive for the right topology. And so in expectation, this thing actually turns out to work out. That is, for every set of 4 species, you get a positive expectation for the right thing. And so this was not proved by us, it was proved by Elizabeth Allman and colleagues. And so that tells you in particular that you get a consistent method. If you have a large number of genes and you do this, you can get your tree. Now the question is, well, how well does it do in terms of sample complexity? That is, how many genes do I need to get a good answer? There are several methods for which we actually know the answer. But this one was open. And if you look at this one again, it's sort of counterintuitive because it really looks like this is going to be a bad method. If you care about the sample complexity, you somehow care about the variance of your distance estimate, right? So I'm showing you that the expectation of this distance satisfy this 4-point condition and therefore give me the tree. If I want to apply that on a number of samples, then there's going to be some variance around my expectation. And it looks here like the variance is going to be bad. If I look at this particular example here, if I take A and B far apart in this long linear tree where I have a sort of an alternation of short and long branches, then it's not very hard to see that the variance of your distance estimate between A and B is going to scale like n the number of species. And that's bad because it suggests that you're going to need a number of genes that are going to scale with the number of species, whereas other methods, I'll come back to that in a second, other methods have been shown to behave a lot better in that respect. Why is that the case? Right? If I just think about what happens in this alternation of short and long branches, this is the short branch means that when I have these 2 lineages coming in, there's a high probability that they won't actually meet here and meet later on, which is what happens here. And now, if that's the case, then there's sort of 2 cases. So either these 2 lineages meet first and then meet this one, or this happens, and then I have sort of an extra edge in my number of edges between my 2 species A and B. So that has a large probability of happening. It creates a lot of variance. And then this long branch here means that everything is going to coalesce, and I'm just going to repeat this thing more or less independently from each of these. And so there's going to be sort of a linear contribution to the variance. But despite the fact that you have this sort of bad variance, it turns out that these estimates are all highly correlated. The difference between A and B is very correlated with A and C and so forth. And that correlation somehow really buys you a lot. Because at the end of the day, what you care about is not the distances themselves, but again, is this 4-point condition. When you plug them in, what happens there? And so this is the best result we have so far. We're still trying to improve it. You actually get a logarithmic sample complexity in n divided by this f here is the length of the short edge. So that measures how much incomplete lineage sorting you have. The best thing known for this other method called ASTRAL, I told you about ASTRAL in the context of gene duplication. You can apply the same thing here to incomplete lineage sorting. And there we can show that it's log n over f squared. So we're not quite there yet, but I suspect that you can get pretty close to that with this other method, NGST. And so in 1 minute, what's going on. And so I said that the correlation really helps you here. If I go back to take my 4 species and I apply my 4-point method, because that's really what I need in order to reconstruct the tree there, you get all this cancellation, right? Because I take the sum of this and I subtract and so forth. That was the whole point of the 4-point condition. In particular, all of this variance comes from this long path here, and it cancels out when I subtract the 1 from A to Z n minus 2 and so forth. And so you have a lot of cancellation that helps you. Now that turns out not to be quite enough. Once you've taken that into account, you still have to worry about the fact that there's a lot of species that creates a lot of different lineages that might all merge with your thing. But if you lose some properties of the coalescent process, one nice thing that it has is that if I take 4 species, what I care about is how many lineages sort of hit the path that's sort of in the middle path of their quartet. And if you think about how the coalescent process works, you can show that even if you have a lot of lineages coming in, most of them sort of merge within themselves, and there's actually more of a logarithmic number that will hit the thing that I care about. And you put all this together with some more tricks, you end up with this logarithmic or polylogarithmic bound on the sample complexity. And so this was just sort of some examples of theoretical results that we've had in the last few years. One thing that people really care about in the literature these days is just to combine all of these into one, because all of these sort of happens in the same example. And so what you want is to have a method that works well for duplications and losses, and also incomplete lineages, as lineage sorting, or things like lateral gene transfer that happen. And so we are looking into combining these, and now you have these much more complicated models. But the nice thing about these methods that I've shown you is that Astral, for example, the one that takes this quartet majority vote, actually works separately for these things, and so combining them together should give you good answers too. And then a more interesting question, maybe from a theoretical point of view, is, we've ignored that first step where I go from the genes, I build the gene trees, and from there I assume that they're correct, and then I reconstruct my species history. But of course they're not correct. My genes have various lengths. Some of them have a lot more signal than others, and there's a lot of mistakes that I make in these reconstruction of gene trees. And capturing, what are the errors that maximum likelihood does in this reconstruction is hard. And so from a theoretical point of view, what we've been doing is to think about developing methods that sort of go directly from the sequences to the species tree and try to understand the sample complexity, which is now a sort of a two-dimensional thing, because I've got the number of genes and also the length of each that affect how much signal I have. But that's sort of an interesting area for future work to better understand these. And so this was joint work with Tandy Warnow at UIC and her student Erin Malloy, and then my two students at UW-Madison, Brandon and Harrison. Thank you.